Well, good afternoon, everyone. I'm really excited to be here. And today, I'm excited to share our vision for building out a state-of-the-art informatics platform to accelerate scientific drug discovery. In my talk today, I will focus on four main aspects. First, I'll describe the current state of drug discovery and why we're leveraging human genetic data. Second, I'll provide an overview of some of the challenges that we have encountered in building out one of the world's largest genetics databases. Third, I'll describe how we are leveraging the Databricks unified analytics platform to address many of these challenges. And lastly, I will highlight some key innovations and lessons learned. Let me start with the fact that drug discovery is an extremely difficult problem to solve. In fact, more than 95% of all the experimental medicines that are currently in the pipeline are expected to fail. This is despite the fact that our industry as a whole is spending billions of dollars in research and development. So one of the reasons why the failure rate is so high is that we don't have good drug targets. In other words, for many genes in the genome, we don't know what their function is and how they're related to disease. However, there's a bright spot in all of this. We can leverage human genetics data to significantly increase the likelihood of success. So why is human genetics data so powerful? Well, genetics is one of the best ways to gain insights into the function of genes and how they're related to disease. Interestingly, every single one of us is carrying naturally occurring mutations in our genome. Most of these mutations are rare and don't have an impact on the gene product or on disease. However, there's a subset of mutations called loss-of-function mutations. And as the name implies, these mutations completely disable the function of the gene product. And when you look at the impact on disease of these mutations, it can vary quite considerably. So on the one end of the spectrum, these mutations can be highly damaging and cause disease. On the other side of the spectrum, they can be protective from disease. And we are particularly interested in loss-of-function mutations that are protective from disease. Because what this enables us to do is to design drugs that essentially block the gene product, and thereby they mimic the effect of such loss-of-function mutations. And our hope is that we could replicate the same protective effect where we get protection from disease. Now, one such example where human genetic evidence played a key role in drug discovery is PCSK9. In early 2003, a group of researchers looked at individuals who had extremely high levels of bad cholesterol and were suffering from debilitating heart disease. And what they found was a gain-of-function mutation in the PCSK9 gene. A few years later, another group of scientists studied the extremes of bad cholesterol in a population. So they not only looked at individuals who had extremely high levels of bad cholesterol and were suffering from disease, but also individuals who had extremely low levels of bad cholesterol and were protected from disease. And what they found in these individuals who were protected from disease was a set of loss-of-function variants in the PCSK9 gene. And what was striking there was that these mutations reduced the risk of heart disease in these individuals by almost 90%. These discoveries led a tremendous amount of efforts by a few companies to develop drugs to essentially inhibit PCSK9. And these drugs have been shown to be effective in the clinic, and some of these were approved later on. Now, this particular success story, together with Regeneron's long history of using genetics-based science to supplement its drug discovery program, the company founded the Regeneron Genetic Center in early 2014. Now, our goal at the RTC is to essentially replicate what we have done for PCSK9 and now scale up these efforts. And to that end, we're building one of the world's largest human genetics databases. Today, we have sequenced more than 300,000 individuals. And at the RTC, we have more than 60 different partners. And we are pursuing a number of different study designs, ranging from small-scale family-based studies all the way up to large-scale population studies. And for the rest of the talk, I'll focus on these large-scale population studies. Recently, the RTC announced a major collaboration with the UK Biobank, where the RTC committed, together with its partners, to sequence more than 500,000 individuals over the next 3 to 5 years. This is truly a massive effort and will provide an unprecedented resource to gain insights into the function of many genes and how they're related to disease. Now, in order to sequence that many samples, the RTC focused on automation from the very beginning. Now, we have the capacity to sequence more than 300,000 individuals per year, which makes us one of the largest sequencing shops in the world. This is great news, but it also creates a huge challenge for us because we have to then be able to analyze all of this data and extract insights. And our hope is to use all of this data to identify new potential drug targets. Now, over the next few slides, I want to describe in a bit more detail how we're actually conducting these large-scale analyses. The approach is actually quite simple. What we're doing is we're sequencing a very large number of individuals, and we identify all of the mutations in their DNA. And then for these same individuals, we obtain de-identified electronic health records from our collaborators, where we then can extract disease traits that we then can further analyze. So once we have these two pieces of information, we can essentially build two different matrices. One matrix is what we call the mutation matrix, where the different rows represent the different mutations, and then going across in the columns, we have all the individuals that are part of the study. And then each cell in the matrix essentially represents, or in there we can indicate whether a certain individual has the mutation or not. And similarly, we can build out a trait matrix, where the rows represent the different disease traits, and then going across the columns, we have the same ordering of the individuals, and we indicate whether a certain individual has the disease or not. What we then can do is, once we have these two matrices, is essentially perform an all-by-all association test. Essentially, what we're doing is we're looking for correlations between every mutation and every disease trait. And we want to see if there's some sort of statistical signal. Now, this process produces a vast amount of association results, which we then have to analyze and mine to gain insights. Now, the picture here that I'm describing on the right is the desired approach. However, in reality, things are quite a bit more complicated due to three reasons. One is that the data that we have is not unified. It is highly decentralized and stored in many different formats. Also, the data itself is organized in very different ways. For example, some of these matrices are not completely squared off. They're transposed of one another, or the ordering of the individuals in these matrices is not the same. So what ended up happening is we spent a lot of time just getting the data ready for analysis, trying to find the latest and all the required inputs to pursue an analysis, and this was just very time-consuming and inefficient. The second challenge is that we have data from multiple cohorts. Often the data that we have from various collaborators is not perfectly consistent. So we have to find ways to unify the data from these different cohorts and be able to aggregate the data. And the third issue is related to scalability. If you look at the input matrices that feed into this process, they're already quite large. So if you look at the mutation matrix, we have on the order of tens of millions of rows in there. And then on the trait matrix side, we have tens of thousands of rows. But then what you're essentially doing is creating a cross-join, and then you end up with tens, if not hundreds of billions of rows in terms of the association results. And the association results is what we want to be able to mine and analyze to gain insights. So in order to address these challenges, we established a major partnership with Databricks. And we are now, the RTC is now using the Databricks Unified Analytics platform to not only unify the data, but also to unify the underlying tools. And together with Databricks, we have been focusing on 3 development efforts. One is implementing and developing highly efficient and unified data representations. Second, implementing highly scalable production workflows so that we can analyze billions of rows. And third, creating a unified code base so that we can enable everyone within our organization to perform compute on this data. Now, one of the key requirements that we had from the very beginning was to make this data accessible to everyone within our organization. Because we have many scientists at Regeneron that know a great deal about the biology of many genes, so we want to be able to make sure that they get access to this data so that they can accelerate their research. And the setup that we're using is actually quite simple, which is shown here on the diagram on the left. Essentially what we're doing is for these web apps, we have an EC2 instance where we run the web application, and then we use Databricks as the backend cluster. So essentially what happens is the web app makes an API call to the Databricks cluster, submits the query. The query then gets executed on Databricks, and the results are returned to the web application, where you then can visualize the results. Now, this architecture has the main benefit of having the same back-end infrastructure. So it doesn't matter if you do an analysis within a Databricks notebook or using a web application, you're guaranteed to get the same results. Now, one of the key applications that we focused on from the very beginning was what we call the RTC Results Browser. The goal of this tool is to be able to, or enable users to search all of our association results across multiple cohorts. Typically, the user would enter a mutation, a gene, or a disease of interest, and then be able to obtain all the association results. Now, what's interesting here is that the underlying data is not static, because we often get data from new cohorts, and we want to essentially append these association results to the existing data set. And we now have more than 67 billion association results in there. And we are now processing or doing the latest refresh, and we're expecting to have more than 200 billion association results in the next refresh. So now, in order to make that work, we had to optimize the process at multiple levels. So one optimization step that we performed is just the ingestion of the raw association results. Now, what I'm showing you on the right here is a graph that shows on the x-axis the number of rows that we have in a particular data set or different refresh points. And then on the y-axis, we have the number of hours or days it takes to process the data. And what you can see from this chart is that for the first 2 refreshes, we actually had a non-Spark solution. And it took days, if not weeks, to process all of this data. And this solution was based on a single machine architecture, and clearly that didn't scale. And so we had to explore other options, and that's why we started to use Apache Spark. So for our first test case, we ingested about 30 billion association results. And then for our latest refresh, the data size essentially doubled. But the compute time is still less than 24 hours. So essentially, the Spark-based solution scales linearly with the cluster size. In addition, we performed a number of optimizations to make the process even more efficient. And this enabled us to move other processes into this overall workflow. And now we have an end-to-end Spark solution, which is much easier to maintain and to automate. The second set of optimizations that we performed is around the data partitioning. So now when we originally ingest the data, it's organized or partitioned by cohort. In other words, it's not optimized to perform queries, because most of our queries are either by a gene of interest or a mutation of interest. So in order to address that particular problem, we decided to implement what we call genomic ranges to facilitate these types of queries. Now, one naive approach is to essentially take the entire genome and then split it into equal bins. However, that approach doesn't work because of two reasons. One is that the genes in the genome are not distributed uniformly, and secondly, the association results within a gene are also not distributed uniformly. So what we ended up doing is creating a process that creates dynamic genomic ranges that ensure that the data partitions are uniform and optimal in size. And by using these optimizations, we were able to reduce the query response time from more than 30 minutes to less than 3 seconds. Now, this type of improvement is quite amazing and has a lot of implications for our web applications, because now with a response time of less than 3 seconds, we can make the web application much more interactive, and users can explore the data in multiple ways to gain new insights. Now you can also use the Databricks notebooks to explore this type of data. And what I'm showing you here is, in the first line, you essentially can load all of this data in just one command. So you not only get the data ready and prepared for analysis, but you also get a set of functions that we have implemented so that you can manipulate the data very easily. So this is truly quite transformative, because before we spent hours, if not days, to get the data ready for analysis, and here essentially gets served up on a silver platter, and we could start right away pursuing the analysis. And then on the second command, I'm showing you the number of association results we have in this data set. And indeed, we have about 67 billion rows in there. And we are quite certain that essentially all the new potential drug targets are somewhere in there. We just have to be able to find and identify them. In fact, the RTC recently identified a new potential drug target for treating liver disease. And specifically, what we found was a loss-of-function mutation in a gene called HSD17B13 that is protective from chronic liver disease. The outside press has called this the PCSK9 of liver disease. So now, over the next few slides, what I want to show you is how we can recapitulate the findings that are in this paper. So when we study liver disease, essentially we can measure two different enzymes in the blood. And for the purpose of this discussion, these enzymes are called AST and ALT. And if you have elevated levels of AST and ALT, they're indicative of liver damage. So what we want to be able to do now is find loss-of-function mutations that are protective, or in other words, they lower the levels of AST and ALT. Now, what I'm showing you here is what we call a Manhattan plot for one of these liver disease traits. And in this particular example, I'm showing the Manhattan plot for AST. And again, we can create this plot just using one line within a Databricks notebook, which is quite powerful. Now, what this plot shows you is essentially all of the association results that you get for this particular liver disease trait. And on the x-axis, the different colors represent the different chromosomes. And essentially, every point here is a mutation that's associated with that trait. And then the y-axis shows the level of significance for that particular association. Now the question is, which one of these mutations is most interesting? And it turns out to be not the large peak, but rather this small peak here. These are the mutations in HSD17B13. Now we can dive a bit more into the details to see what's so interesting about these set of mutations. What we can do is perform a query within Databricks Notebook to get all the results for this particular gene, HSD17B13. Again, this is a one-liner. We can then further subset the results just to the Geisinger cohort and order all of the results by p-value. Now, Databricks makes this, or the code in Databricks makes this very simple. And when we perform a count, we see that we have roughly 3,700 association results. Now, what we can do is drill down a bit more and see what's so interesting here. So what we're doing here is essentially narrow the results to liver disease traits, and we're also specifying the mutation of interest. And we get this output table that's shown here. Now, there's two key takeaways. One is that this mutation is associated with a broad spectrum of different liver disease traits. And secondly, all of these associations confer protection from liver disease. So what you can see here is that AST and ALT essentially are associated, or they're lowering. This association lowers the levels of these two enzymes. And you also see a protective effect for alcoholic liver disease, cirrhosis of the liver, and so forth. So now the Databricks Unified Analytics platform makes it much easier to explore and mine this data in new and different ways. And as you probably can tell, we only started to scratch the surface in terms of how we can analyze and mine this data. And I think there's a lot more to come. So in conclusion, at Regeneron, our goal is to bring the power of science to medicine and develop new drugs for patients in need. At Regeneron, we strongly believe in the power of leveraging human genetics data in the drug discovery process. And to that end, we're now building out one of the world's largest human genetics databases. We have been extremely fortunate to have a great partner in Databricks, and together we have essentially created a state-of-the-art data science platform from scratch. And as part of this collaboration, we have been focusing on three main areas: developing highly efficient and unified data representations, building out highly scalable workflows to mine billions of rows, and we also addressed some of the key bottlenecks there. Specifically, we have been able to reduce the ETL time from weeks to just a few hours now, and we were able to optimize the query response time from more than 30 minutes to less than 3 seconds. We also have built a unified code base within the Databricks notebooks so that we can enable all levels of users within organization to perform compute. And most importantly, the Databricks Unified Analytics platform not only brings together the data, the tools, and the people, but it also enables us to accelerate innovation. So with that, I would like to acknowledge everyone involved, and I would be happy to take questions.
Step to the mic, or I can pick it to you if you'd like. Any questions?
Hi, thank you for the great talk. So my question was related with the traits. And the traits, I mean, do you mine it from known PubMed literature? Or because that's a long research paper itself, like how do you associate a gene with a certain trait? So how do you pick up that part of the literature, and do you rely on that through PubMed?
Well, so that's a great question. So the way these analyses are structured, right? We have at the RTC we have multiple collaborators, and we're essentially doing the sequencing in-house. But then we get the clinical data, the de-identified electronic health records, from our collaborators, and then from these de-identified EHRs, we essentially derive a set of disease traits that we then use for the analysis. So we don't have to mine the literature. We get this information from our collaborators.
I have a question as well. In the beginning of the talk, you were talking about sequencing the individuals and identifying their mutations and not having background in the space. How do you identify, what are you measuring the mutations against? What's your baseline that you determine if someone has a mutation of a gene?
So what we are doing is we're using exome sequencing. Essentially, we are sequencing the protein coding regions of the genome, and there's a sort of a well-established process to do the sequencing and then also to perform the downstream analysis. Essentially, what we're doing is from the sequencing instruments, right, we get a set of sequencing reads, which are then aligned to the reference genome, and then there's established algorithms to call the mutations. So essentially, that's a great question. Essentially, it is a sparse matrix because most of the mutations in the genome are extremely rare, and so I would say a sparse matrix lends itself extremely well to represent this type of data.
So so my question is, so what are the next set of sort of challenges that you guys are facing now that you've solved those three you've talked about using Databricks? What is the next step function for you guys? Is it just acquiring more data from different partners, or like, you know, what's in the roadmap right now for you guys?
We are certainly, you know, continuing to sequence at a very rapid pace, and are, you know, analyzing many more cohorts and more larger cohorts as well. And I would say now that the challenge is to, you know, find new and innovative ways to mine all of these association results to identify new potential drug targets. And we have a number of ideas to do that.
Cool. Thank you.
And then we'll get to you.
Hi, thanks for the talk. I was wondering, as your lab techniques improve over time, have you faced batch effects from that? And if so, how have you worked with it in your data science department?
Excuse me, could you?
So as your lab techniques have advanced, new capture kits or whatever, have you faced batch effects from that in your data? And if so, how have you dealt with it?
Well, I would say because we're doing all of the sequencing in-house, right, we have a highly automated process to do the sequencing. And by using this automated and robust sequencing process, we have been able to virtually reduce all of these batch effects. So that's one great benefit of doing all the sequencing in-house, where we have full control over the entire process.
What kind of tools are you using to generate the association results?
So right now, what we're doing is we have an established pipeline or an engine for producing these association results. We are currently using mostly Plink to get these association results, but now we're exploring different options in Spark as well to do all of the association testing using this framework.
So just following up on that, have you looked at Hail to do more Spark-based, even the association studies?
Right question. So we are certainly watching Hail quite closely, and we have looked at the Hail code base, and I think it's a great project. However, it doesn't really work that well for our particular use case, because Hail is mostly designed to deal with one cohort, and we have more than 60 different cohorts. So there's a lot of additional things that need to happen, and managing all of the samples across multiple cohorts is critical.
This will be the last one.
So my question is, how do you make the leap from once you identify the mutation to reducing costs for drug discovery?
Well, the RTC is, the subsidiary of Regeneron, and we are highly integrated with the rest of Regeneron. And that's one of the beauties, because once we have a compelling finding using our data, we can then sort of do the next step right away, where we can do functional modeling and see, or sort of doing a deep dive into the biology to see whether we can validate this finding. So I think it's a big benefit of being part of this integrated approach, where we can, where we have all the tools available to sort of take it to the next step.
Okay, thank you.
All right, guys, let's give Dr. Habegger another round of applause.